interaction with the FDA, the ability to discuss with the FDA surrogate endpoints and intermediate clinical endpoints in order to go along an accelerated approval route. And there's unique to the regenerative medicines, the ability to satisfy full approval, post-accelerated approval with not so standard approaches. Standard approaches are usually a randomized clinical trial, phase 4 clinical trial to get full approval. In the RMAT designated cases, they can use real-world evidence, patient registries, data from a limited number of sites that are, that's basically, like I said, unique to cell and gene therapies or regenerative medicine. Now, this is only, this act has only been around a couple of years. I think there's about 30 or 40 RMAT designated products right now. Since none of them have been approved, the accelerated approval theoretical paradigm hasn't been gone through from a practical perspective yet, but that's the concept.
So when you say real world evidence, are you talking about supplementing it post-approval, or are you talking about during the clinical development pathway, the process is different?
Post-accelerated approval, you need to do confirmatory trials usually to get full approval. And in the cell and gene therapy RMAT designated case, that requirement, that data requirement is a lot more flexible and not as stringent.
Okay.
And you can use different types of evidence.
And do you see any difference? So, you touched on the fact that many of these therapies are intended to be curative in nature and kind of single administration. Do you see any difference in terms of long-term safety data that's needed, or is this something that's covered as part of this post-accelerated approval aspect? Or, in terms of if you get an IND filed in gene therapy versus an IND in biologics, is the path to getting potential approval faster because of the RMAT designation, or is there a need for longer safety studies because of the fact that it's a theoretically one-time curative treatment?
So they're not coupled. So RMAT designation might allow for approval first, but the long-term safety follow-up is independent of that. So even after approval, for example, I believe the Novartis CAR T product, Chimera, negotiated, it was either a 10 or 15 year follow-up requirement for all patients treated. You would never get that requirement in the small molecule or biologic space, other biologic space. And the reason for that is we simply don't know the impact of a gene therapy or gene within a cell, that therapy, that long-term impact. So pretty much those long-term follow-ups are going to be required either way. They're not a hindrance to approval unless there's data to show otherwise. But from a theoretical perspective, every company in the near term that has a gene therapy approval will have to go through similar requirements for long-term follow-up.
Okay. And as far as the RMAT designation is concerned, is it safe to assume that all gene and cell therapies in theory would qualify for that, or are there additional requirements in terms of indication or unmet need or other aspects of the filing that would be needed to check the box to get that designation for a gene or cell therapy?
Yeah, so there's 3 requirements. Number 1 is you have to meet the definition of a regenerative medicine product, and it's contained with, there's a guidance on this, a draft guidance that you can look up in Google on expedited programs for regenerative medicine products, and the definition is in there. The second requirement is the clinical indication has to be serious / unmet medical need, similar to the other expedited pathways. And